look carefully. This is a cell and it's going to be divided into two, right? So look, this cell we consider, or just let's come in another way and know it. This cell is going to produce two cells, right? When it produces two more cells, where does this cell go? What happened to that cell? So my dear friends, actually this cell and this cell, both of them are same, but this only one new cell has been made, right? Only one new cell has been formed. It is there that you have marker in your hand, right? This marker, and this marker is going to make a copy of itself. This marker is only one marker and it is going to be divided into two markers. Both of them are same. You can see both of them are same. So how many new markers are formed? Only one. Only one. And this was the old one. So during mitosis, only one new cell is formed and the other cell is our previous cell, right? So this cell is actually going to copy itself one time. So whenever this cell will copy itself, it will make only one cell. So actually there are two cells formed, but this one cell is our old cell, which was going to divide, which was going to make a copy of itself, and this is the new cell which is formed. So during mitosis only one new cell is formed and the other cell is our old cell which was dividing, which was the parent. Clear? Done. This concept is clear, I hope. Now we want to know that these chromosomes have been created 92 chromosomes. The 46 are the older ones and these new 46 are formed. How these are formed? These are formed during interphase. These are formed during interphase. So let us write a little detail of this cell which is going to divide. Clear? Let me move forward. Friends, if this is my cell, that's this one, and here are many cells which have been damaged, and around those cells there are healthy cells which are going to divide, which are going to make new cells. So they have to make the extra copies of the DNA, right? So look carefully. Here in this cell there are about 46 chromosomes. Clear? How many chromosomes? 46. So we say that 23 of them have come from my mother side and 23 of them have come from my father side. So total makes 46 chromosomes, right? So we say that one small n has come from my father and one small n have come from my mother, and this small n is equal to 23 chromosomes. Clear? So how many total chromosomes we have in our cell? We have 46 of the total chromosomes, right? So look, we say that 1n plus 2n is equal to 2 small n, 1n, 1n, 2n. So from 2n we mean a diploid. Diploid. Clear? So diploid means 46 number of chromosomes. Diploid means 2 small n. Clear? Done. I'm writing the detail of this cell, and what will happen to this cell here, we will see that, right? Look carefully. Then the amount of DNA which is present in chromosomes, and here let me make the chromosomes also, right? For example, this is a chromosome from my father's side and this is a chromosome from my mother's side. Clear? And both of them have their DNA inside. This is the DNA. So dear friends, one capital N DNA comes from father side and one capital DNA comes from my mother side. So I have one capital N from father, one capital N from mother, and total of 2 capital N, right? So capital N shows amount of DNA, right? So the DNA which is present here is 2N. And the number of chromosomes which are present are 46, right? So this small n shows number of chromosomes. Clear? Small n shows number of chromosomes and Capital N shows amount of the DNA. So this cell is 2 small n and 2 capital N. 2 small n means it has 46 chromosomes, and 2 capital N means it has amount of DNA which is equal to 2n. Why? Because 1 in DNA came from my father's side and 1 in DNA came from my mother's side. Now look, what will happen? This cell has decided to enter to mitotic phase or division phase. Clear? As we know here from cell cycle, I had told you that cell cycle comprises of how many phases? It comprises of 3 phases. 2 phases, right? First was from here up to here, whole this process was interphase, right? And it has G1, S and G2. Clear? And then from here up to here, this was our M phase or mitotic phase. So there was prophase, prometaphase, metaphase, anaphase, telophase and cytokinesis, right? So we are actually discussing this M phase. Now look, listen carefully. This cell that I have discussed right now here is present here. Clear? And it has how many chromosomes? 46. Done. Now when this and the chromosomes which are present here are like this, and you know during S phase what happens? DNA replication. DNA replication. I mean DNA is double. So when the cell comes like this during interphase, what happens? That when it reaches here, the number of chromosomes are same, but the chromosomes have become like this. Right? So it means that this chromosome has 1 DNA, 1 in DNA, 1 capital N DNA, and this chromosome has 2 capital N DNA. So look, during interphase what happened? The amount of DNA is doubled. The chromosomes from single structured have become double structured. Look, this chromosome which has only 2 chromatids on each sides of centromere, this is centromere, right? And this is chromatid. Clear? So it has DNA in itself, in its chromatids, right? And that chromosome, when it becomes like this, so what is the difference between this and that? The difference is it has only 1 chromatid on each side of the centromere, and this chromosome has how many chromatids? 2 chromatids on each side of the centromere. So we call this chromosome as single structured chromosomes, and this chromosome as double structured. So double structured means that it has 2 capital N DNA, and single structured means it has 1 capital N DNA, right? And how many chromosomes are present here? So you have to count the number of chromosomes from the number of centromeres. So how many centromeres here we have? Only 1. So number of chromosomes 1. And how many centromeres do we have here? Only 1. So number of chromosomes are 1. Chromosome number is same, but the amount of DNA is double. So you put in your mind that whenever the cell enters to interphase and it comes out, it has the same number of chromosomes, but the amount of DNA is doubled. Done? Okay, so up to here, I hope you have understood all what I have told you, right? So let us see that whenever this cell wants to divide, this cell must enter to interphase. This cell has gone to a phase which is called the interphase. So here we did our interphase. So whenever the cell comes out of interphase, what had happened to that cell? You know that the chromosomes have become double structured. Very good. Like this.